We can grow enteroviruses in culture. We use small tubes and we can look at them by making an aliquot from a stool suspension and inoculating onto the cells and then looking under a microscope each day. And if there is an enterovirus, it can appear to kill the cells, which we call cytopathic effect. And that's where actually the cells are destroyed and that's visible under a microscope. To confirm that we have grown an enterovirus in culture, we can do what's called a real-time PCR. And that shows in real time with the graph where we get an increasing signal shown on the machine. And we have a positive and negative control to confirm that the test has worked correctly. And if we do get a positive signal for enterovirus, then we go on to type the enterovirus. To type an enterovirus, we have a set of strict criteria where we compare it to the unknown sequence, we compare to known prototypes, so they're confirmed enterovirus sequences for the different types. And we have a set of criteria that it must meet, including how genetically related it is, and also what is the percentage sequence comparison to the known prototype sequence. So traditionally, with the enterovirus typing, we've just sequenced a small portion of the enterovirus genome, less than 10% of the genome. But now we want to get whole genome sequencing, as we feel that this will provide us with a lot more information. And like we've heard for the SARS virus, where there's been variants of concern, they've been identified by having the whole genome sequence available. And that's what we now want to do with enteroviruses, to sequence the whole genome and make comparisons in areas that we haven't looked at routinely before in a diagnostic setting. And we think that this information will be very useful for our understanding of enterovirus infections. So the enterovirus whole genome sequencing is in the development phase, and that's where we're having to now try to use new methods. And this is where the fundraising that is being done for this work is so important, that it will allow us to transition from the traditional virus culture to whole genome sequencing. And I really thank everyone for their interest in this work and all the fundraising that has been done by Candice and her family. Thank you.